Okay, we are going to pick up where we left off on transcription. And we start talking about termination of bacterial transcription. So termination is simply just the end of RNA synthesis. It occurs when the short RNA-DNA hybrid of the open complex is forced to separate. This releases the newly made RNA as well as the RNA polymerase and the DNA template. So there are two mechanisms we're going to talk about that occur in bacteria. One is row-dependent termination. It requires a protein. So for row-dependent termination, here we have our DNA template, and we have the RNA polymerase moving along, transcribing this DNA template, and here's the RNA in red. Now, the polymerase will pass by what's known as a row recognition site in the DNA, and then that will be copied into the RNA. So this site in the RNA is called a RUT site, or the row utilization site. Now, this is functional in the RNA, not in the DNA. So it's the copy that has been made in the RNA that is important here. All right. Oops. So what happens here? So the RUT site is a sequence in the RNA where the protein row, which is shown here as a 6-member protein, is a helicase. Here's the structure of row shown over on the right. It will bind to this RUT site and then start moving from the 5' direction on the mRNA in the 3' direction, essentially chasing after RNA polymerase. So it's hydrolyzing ATP and using that energy to move. Now, between the RNA polymerase and the and row, oftentimes you'll get a formation of a stem loop. Now this stem loop is important. It's not a specific kind of stem loop, but what this does is this slows down RNA polymerase and allows row to catch up. When row does catch up, it will cause the dissociation of the RNA of itself, RNA polymerase and the DNA. So everybody is released and RNA polymerase can go reinitiate transcription, the messenger RNA can go be translated, row is then available to go do its termination thing again. Now, there's another form of termination. This is called row-independent termination, or oftentimes it's called intrinsic termination. There are transcription termination sites throughout the genome at the ends of operons. And these are simply inverted repeats. So what does that mean? So it means if you look at this sequence here, this is the DNA sequence. If you look at these blue portions, on one strand, we have AA, GG, CT, CC. That can base pair with these TTT, CC, GA, GG. So it can form an intramolecular base pairing, which will result in a stem loop. So the portions in blue can form hydrogen bonds and they will base pair. The part in the middle here does not form, and that's our loop. So we have a stem and a loop. Now, the stem loop was important in row-dependent termination, which I just showed you. But this kind of stem loop that's involved in the intrinsic termination is slightly different. It has these uracil residues at the 3' end. Right? So it's a uracil-rich sequence at the 3' end. And this is important for termination. So same kind of things happening where RNA polymerase is transcribing a region of DNA. The mRNA here in red is being made. Now there's this stem loop will then form once the RNA polymerase passes through the region that codes for it. Now there's a protein NUS-A which helps to stabilize RNA polymerase, so this causes a pause. Now inside the polymerase, this is the 3' end where all these uracil residues are. So these are going to form weak hydrogen bonds with the DNA template. This is going to make it easier for RNA polymerase to dissociate from the DNA template, releasing the RNA itself and the DNA. Okay. Now, no protein is required for this. NUS-A can help, but this can happen without the action of NUS-A. So that's why this is called intrinsic termination. It's just intrinsic. The termination function is intrinsic to the sequence of the RNA. Okay, so I have a short video which I will play for you, and this sort of sums up all of bacterial transcription. Okay, so that's a really nice summary of prokaryotic or bacterial transcription. So now we'll move on to eukaryotic transcription, which, as in most cases, is always more complex than its bacterial counterpart. So eukaryotes possess 3 different RNA polymerases, and they transcribe different classes of genes. And keep in mind that transcription occurs in eukaryotes in the nucleus. For bacterial cells, it occurs in the cytoplasm, as there are no separate compartments within a bacterial cell. Okay, so RNA polymerase 1 mostly transcribes ribosomal RNA genes. RNA polymerase 2 is a very interesting one that we're going to be talking about. So this is one we're going to focus on. This transcribes all protein-coding genes, microRNA genes, and some other non-coding RNAs. RNA polymerase 3 transcribes smaller RNAs, transfer RNAs, the 5S ribosomal RNA gene, and many other smaller RNAs. So for comparison, here are our 2 promoters, a schematic of 2 promoters. There is on top, the eukaryotic promoter, shown here, and down here we have prokaryotic promoter, which I've shown you before. And so they have similarities in that there are promoter regions. This is where RNA polymerase is going to associate with the DNA to start transcription, and we have promoter region that we talked about in bacterial cells. And then there are regulatory sequences, oftentimes in bacteria upstream of the promoter, and the same thing occurs in eukaryotes. Usually the location for these kind of elements are a little bit further than what's seen for bacteria. And you can also have elements located not just upstream, but also downstream. And you see that very occasionally in prokaryotic genes. And the transcription will start in the same sort of numbering system where transcription starts +1. So everything that's being transcribed, these numbers are increasing, +1, +2, etc. And to the left or upstream of the transcription start site, the numbers are negative. Okay, so for transcription initiation to occur in eukaryotes, it's slightly different. Now, first thing I want to mention is that I'm going to start using some abbreviations. So RNAP2 just refers to RNA polymerase 2 from eukaryotes. And then we have these TF proteins. And so, for example, TF2D is transcription factor for RNA polymerase 2. So TF2 and D is just probably in the order that they were discovered. They were given letters. Okay, so what goes on here? So TF2D is a very important factor. So this is the factor that recognizes the promoter for eukaryotic genes. And so the promoter is slightly different. There's a thing called a TATA box, which was shown on the previous slide, and that's bound by a particular protein that's part of TF2D. So TF2D is a multi-protein complex. What we're going to focus on for this is that the TBP, or TATA binding protein, will recognize the TATA box and bring all these other factors to this location. So we're still not at the point where RNA polymerase comes around. Another transcription factor, TF2B, binds to TF2D. And this promotes the binding now of RNA polymerase to this promoter. RNA polymerase brings along with it TF2F, and then other factors are recruited, TF2H and TF2E. Now this whole pre-initiation complex here is a closed complex. We've talked about closed and open complex. The promoter is still double-stranded and hydrogen bonds are formed in this region. All right, so there's some interesting things that happen here. TF2H is a helicase, is going to start unwinding DNA. It also starts to add phosphate groups to the carboxy terminal domain, that's what CDT stands for, of RNA polymerase 2. And this will help to break contacts between some of the other factors, and you'll get release of those factors. All right, so this is actions of TF2H. After these factors get released, now the promoter DNA becomes unwound, and we get this open complex formation, and we start to get synthesis of RNA. Note that on the polymerase, phosphate groups have been added to the polymerase. So now RNA polymerase at this point can proceed to the elongation stage. There's been initiation of RNA synthesis, and it's going to escape from this promoter and start synthesizing RNA. And the main thing to remember here for transcription elongation is that it's pretty much the same thing that's going on in eukaryotes that goes on in eukaryotes is happening in prokaryotes also, that we've already talked about. There are some subtle differences that we're not going to consider for this class. So if we need to start initiate transcription, we're going to elongate transcripts, and then we need to end transcription in eukaryotes. And so how does that occur? So there are a couple models for how this occurs. So we're talking about RNA polymerase II. It's going to transcribe past the polyadenylation signal, and we're going to talk about that sequence and what goes on there in a little bit. But suffice it to say that the RNA is cleaved just past this sequence. So it's there's a cleavage releasing the messenger RNA part, so this part here, while the RNA polymerase can continue to transcribe. So one model is that this sort of cleavage and going past the signal sequence, it just sort of destabilizes RNA polymerase II. It dissociates from the DNA. There can be release factors, release of elongation factors, or binding of other termination factors, and they're not shown here. So this is when termination occurs. This is called the allosteric model. There's another model, which is the torpedo model, and with that, the idea here is that after the messenger RNA portion is released, there's this bit of transcription that continues. Well, this can be a substrate for an exonuclease. So we talked about exonucleases and endonucleases, how they are enzymes that can break bonds in DNA or RNA, so in nucleic acid. And this would be a 5' exonuclease. It would start cutting nucleotides off of the 5' end of the RNA. So this enzyme basically follow behind the polymerase, degrading this RNA portion here. And there's a lot of details that we have not gone over here, and some details still need to be worked out for these systems. Okay, so another thing that's very different from prokaryotic transcription is that the mRNAs that are made in eukaryotes undergo a great deal of processing. And so I'm not going to mention everything here. This is a nice table from your book that sort of summarizes what's happening in these, or can happen to RNAs during the transcription process. And we're going to go through some of these in a bit more detail in the following slides. So this kind of processing called post-transcriptional processing. So this and this is for messenger RNAs. Happens, as the name indicates, after transcription, but that's not completely true. These start to occur as transcription is proceeding. So this occurs in the nucleus, where RNA polymerase is going along and transcribing. So there are a few different modifications. One is the addition of what's called a 5' cap. Again, we'll talk about that. There are sequences in the RNA that don't make it into the final mature messenger RNA called introns that need to get removed by a thing called intron splicing. And then this poly A tail that I mentioned will get added on to the 3' end of the messenger RNA. Now, the factors that are involved with doing these different things are associated with the RNA polymerase carboxy terminal domain. And as I mentioned before, this is the RNAP2 or CTD for carboxy terminal domain. So the factors are already on the polymerase. so that as the messenger RNA, shown here in sort of darker blue region, as soon as the substrate becomes available, so the 5' end in this case becomes available from the RNA polymerase, the factors that are involved in adding this thing that's called the 5' cap can associate with it and add the cap before transcription of the messenger RNA is done. Okay, so what is this 5' cap? So it's what's called a 7-methylguanosine is added in a 5' to 5' linkage. This has the effect of protecting the messenger RNA from nucleases, because this is a very sort of odd association of a nucleotide on a chain, this linkage. And it's also involved in transporting the messenger RNA out of the nucleus into the cytoplasm. And so it looks something like this. So here we have on the right here, our messenger RNA, and we have this would be the 5' nucleotide, one at the very 5' end. It would have 3 phosphates coming off of it. And so this is sort of normal, and it's linked to the next nucleotide by this phosphodiester bond. But if you look at the 5' end of the messenger RNA, well, there's another nucleotide that's not coded within the DNA that gets added on. And it's a 7-methylguanosine, so it has this methyl group here. So it looks like guanosine, except has an additional methyl group. And it is linked from its 5' carbon, so we've talked about this in previous lectures, to the 5' carbon of the end of the messenger RNA. Normally you have the phosphates from linked to the 5', the phosphate on the 5' end of this nucleotide is linked to the 3' hydroxyl on the previous nucleotide. Here we have going the connection is from a 5' carbon to a 5' carbon. So this is very odd sort of structure here, and it's often not recognized by many of the nucleases that are involved in degrading messenger RNA. So it protects the messenger RNA. All right, so now we're going to talk about the other process, introns. So introns are called intervening sequences. They exist in the original RNA transcript. They're encoded in the DNA, but they don't make it into the final messenger RNA, and the sequences within them will not get translated into any amino acid sequence of the final protein that's made from that messenger RNA, because they are removed from the messenger RNA. Now, there are going to be portions that code for the protein, and we'll mention those in a moment, that will get put together, they'll get spliced together, introns will be moved out. Those portions are called exons. So exons are the regions that stay in the messenger RNA, will code for a protein. Introns are those that are initially in the RNA but get removed, and the exons will get stuck back together in a particular order. Now, this, if you think about it, the size of the final mRNA, or the mature mRNA, is going to be much smaller than that of the one that is initially synthesized, or if you want to think about the gene that is encoded within the DNA, because there's a bunch of sequences that can be removed, and messages, so genes can have more than one intron. Now, this was originally thought to occur only in eukaryotes, but this is not true. Introns occur in bacteria. They are self-splicing introns, which we'll mention in a little bit, discovered by David Shube at the University of Albany, and independently by Jeffrey Palmer at, I forget where he was, at the University of Illinois. Okay, so what is splicing in a sort of graphical, schematic sense? So here we have what's called our pre-mRNA. So this is the sequence of the RNA that corresponds directly to what was in the DNA, and we'll have these regions in blue, which code for a protein. These are functional. So we have an exon, what's called exon 1. Here then we have an intron. So this is not going to make it into the final messenger RNA. This will get removed, and then downstream of that, we'll have exon 2. And you can have more than, and you can have another intron down here and more exons. And you can have many introns and exons. But if we're looking at just this case where we have these 2 exons that are broken up by this intron, the intron gets removed, and the 2 exons get put together in this order. So this would be the mature messenger RNA, and then there will be an additional RNA that has the introns. No known function has been shown for it, like these introns by themselves. Right, so this process of removing the introns and putting the exons together is called splicing. Now there are 3 different splicing mechanisms that have been identified. So there's group 1 intron splicing, group 2 intron splicing. These can also be self-splicing introns, so they don't require any proteins. The intron can do everything by itself. And then there are spliceosomal introns, which we're going to talk about first. And these are lots of proteins and RNAs that come together to remove these introns. So again, they are all involved in removing the intron RNA and linking the exons of the final mature RNA together by phosphodiester bonds. So for spliceosome introns, here is a schematic of a spliceosome intron. So there are some things to note here. So in blue here we have our exons, and in yellow we have an intron. And there's a lot of sequence. Most of the sequence in these introns is irrelevant to the splicing. And there's only a few portions that are important. Usually those are right at the junction between the intron and the upstream exon, and the junction between the intron and the downstream exon. And then there's a adenine residue that's important in the middle. And so we have some sequences that are shown here that are conserved, just to remind you. All right, these are the different nucleotides, A, G, U, etc. R just represents any purine, so R means it's either an A or a G. Y means it's any pyrimidine, that it would be a U or a C. And if in DNA, this would also correspond to thymine, so to T. And then N, of course, can be any of the nucleotides. Okay. So this region up here between exon 1 and the intron is called the 5' splice site. The region downstream between the 3' end of the intron and exon 2 is called the 3' splice site. This whole yellow portion gets removed, and these 2 exons get put together. Now this A is going to be important. This is a branch. It's called the branch point A, and I'll show you a little bit more in the next slide, and then in a video this will get talked about also. So here's sort of a schematic of the process of spliceosomal intron splicing, and Richard Roberts and Philip Sharp got the Nobel Prize for working this out. And so here again, just to remind you, in blue we have our exons, and in yellow we have the intron. That branched A that we were talking about is located right here, so that's shown there. Now there are other things on this figure that weren't in the last one. So what are these things? So these guys, this green portion with this little clover leaf structure shown in here, is a SNRP. That stands for small nuclear ribonuclear protein particle. These are contain protein, which is in this case shown as sort of this green, sort of somewhat oval shape here, and the RNA is this little cloverleaf structure here. And the RNA can form hydrogen bonds and recognize bases in different places along the mRNA. Now this U1 SNRP recognizes the 5' splice site, so it can form hydrogen bonds and recognize the sequences that are here. U2 SNRP can recognize the branch point. U4 and U6 are involved in catalysis, and U5 helps to line up the 2 exons. Right? So this whole thing comes together, bringing the 2 ends of the intron together so that the upstream exons, so exon 1 and exon 2, will be close to each other so that they can be joined together and the intron removed. Now, they will, this is an enzymatic reaction, and this splicing occurs. It will release the different SNRPs, and in this case, the U2 SNRP, U4, U6, and U5 will still be associated with the RNA, the intron RNA. U1 goes off by itself, and the 2 exons are put back together. They're spliced together. So this adenine is important because the 5' end of the intron gets covalently linked to this adenine, and it forms this structure called a lariat. So like sort of a lasso that you see in the old Western movies. That's why they call it a lariat. And that's going to be important. That structure is important. Okay, so here's a short video explaining this. Okay, so those are spliceosome introns. So I also mentioned that there are introns called self-splicing introns. So this was a big discovery that RNA could act as an enzyme. It didn't need any protein. It could catalyze reactions. RNAs that do this, and there are multiple kinds of RNAs that do this, were called ribozymes, or an RNA enzyme or catalytic RNA. You hear those terms quite often. Now, this self-splicing mechanism for introns was worked out by Tom Cech, and he got the Nobel Prize for this in 1989. Okay, so let's start with talking about group 1 introns. So group 1 introns are shown here, just to give you, this is from a different book, so the coloration is a little bit different here from the previous examples. The exons here are now in red, the intron is in green, and same setup. We have exon 1 on the left and exon 2 on the right. So what happened here? Now this green portion, this is really a simplified version. This green portion of the intron folds up into a very complicated structure, similar to proteins. We'll talk about translation in a little bit in another lecture. But we've all seen different protein structures that are involved in catalyzing reactions. So the RNA folds up into a structure through various base pairing interactions, intramolecular base pairing interactions, and it will form an enzymatic structure that can catalyze the breakage of the bonds and resealing of bonds. Now, so one of the sites in this structure is a place that will bind guanosine. This is not guanosine that's encoded in the intron or in the DNA or anywhere. This is just a free guanosine nucleotide. It needs to bind to that nucleotide. That's going to be important for splicing because that guanosine is going to attack the 5' end of the intron and become covalently attached. So it was not originally encoded within the intron, but now it ends up at the 5' end. When that happens, this upstream exon, exon 1, has a free 3' hydroxyl, which will attack at the 5' end of the downstream exon, so exon 2, and the two exons will become linked, and the intron will be released, and it will eventually get degraded. Okay. So I've said, and this an important point to make here is that this intron is linear because it looks different from what we just saw for the spliceosome intron, which had this lariat shape. Now for group 2 introns, the intron again is shown here in green, and that again forms up into a different 3-dimensional structure. It does not require this external guanosine. Instead, it has an adenine encoded within it in a very particular location that will attack the 5' end of the intron and become covalently linked. So it's now through an intramolecular interaction. So this branched adenine becomes linked to the 5' end of the intron. And the same process here. There's once that happens, the exon 1 is now free. It has this 3' hydroxyl group. It will make a nucleophilic attack and join up with the downstream exon, and the intron will be released. So you get the mature messenger RNA and this release of the intron. This is a unique sort of association here. It's the actually 2' hydroxyl in the adenine, which is involved in making this attack. But this sort of structure for the intron is a lariat structure, which is the same as the spliceosomal intron. So it is thought that these group 2 introns were precursors to the spliceosomal introns, and that the catalytic function that was contained within this RNA had been broken into different parts and different interactions from different RNAs forming these SNRPs and the catalytic active site. And just the last thing, that both of these kind of introns exist in bacteria. The spliceosomal introns do not. Okay, so a word about introns in general. I said you can have more than one intron in a gene. And if you look at the dystrophin gene, the size of the gene is 2,000 kilobase pairs. However, the mature messenger RNA is only 17 kilobases. most of the sequence in that gene is taken up by introns. There are actually 50 introns in there, and in order to get down to this size, all those introns need to be removed. And you can see that there's the same sort of trend going through all these different proteins. The messenger RNA, is much smaller than what's encoded in the DNA, because there can be several introns, and those introns need to get removed. Messenger RNA is going to be smaller. Now, having so many different introns is interesting, and provides another sort of source of variation in what's coded by genes. So this is a topic having to do with intron splicing called alternative splicing. What can happen in alternative splicing is that you have multiple introns within a gene. Well, some of those introns get spliced out, others may get skipped over. You can get more than one protein coding gene out of the same gene. So the messenger RNA, you get multiple messenger RNAs coming out of one gene, depending on how they are spliced. Large portions will be the same, but there will be differences in amino acid sequences that in these alternatively spliced messages, so that each of the resulting polypeptides will have unique characteristics. Now, the degree of splicing and this alternative splicing varies between different organisms. Yeast, which is a model organism for much of genetics, has about 6,000 genes, and 5% of them encode messenger RNAs that are spliced. And only a few of those are actually alternatively spliced. Now, humans contain approximately 22,000 genes, and this number is changing all the time. Most encode messenger RNAs that are spliced, and it's estimated that about 70% of them are alternatively spliced. And these alternative splicing pathways for some messenger RNAs can result in dozens of different messenger RNAs all originating from the same gene. So we'll talk about an example. The example is the gene that encodes alpha-tropomyosin. This protein functions in the regulation of cell contraction. It's found in smooth muscle cells, striated muscle cells, and it's also found in non-muscle cells at low levels. Now, the different cells in multicellular organism need to regulate how they contract in subtly different ways. And one way to accomplish this is to produce different forms of this alpha-tropomyosin by alternative splicing. So what happens here? So in order for this to happen, so to not have this exon 3, and I'll just use this one example, but it's what's happening at the different positions for this alternative splicing, is that this intron and this intron sort of act as one, and this whole unit gets removed, and 2 is considered the upstream exon, and 4 is the downstream exon, and they will be joined together. Now, how does the cell know to skip exon 3? So that leads us to our next slide, is that there are ways in which the cell can distinguish which exons should be skipped. And so there are splicing factors. So there are proteins that can regulate which exons are either contained within or removed from the final messenger RNA. So splicing repressors bind to the splice sites and will prevent certain exons from being incorporated. So that means this splice site for exon near exon 2 won't be recognized, and this whole unit will get removed. So this is a splicing repressor. It's not allowing this intron here between 1 and 2 to splice properly. It's preventing that from splicing, and instead treating this here from this portion here at the upstream part of the intron, the 5' end here, as the 5' splice site. And now this portion here between the intron and exon 3 is the 3' splice site. So exon 2 ends up getting skipped. Now, if there are splicing repressors, there are also things called splicing enhancers. These can make sure that they can make sure that a certain exon actually gets included and helps to improve the recognition of the splice sites, the splice junctions. So here are enhancers that bind around exon 3. Normally, exon 3 would get skipped, but the production of the enhancers make exon 3 recognized as an exon, and we now get splicing between, get this intron to splice out by itself. The intron between 3 and 4 also splices, so we end up getting 3 incorporated in the final messenger RNA. So that's a just sort of gives you an idea of how much more complex this is. Okay, so the sort of last modification we're going to talk about here is that of the adding on a poly A tail. So we've talked about that the RNA polymerase can come through and make this RNA. So here's the RNA in green. It comes through, synthesizes it. There's a sequence that indicates that the message needs to be polyadenylated. And I'll talk about it and what that means in a moment. So, but I mentioned before that after this, the RNA polymerase passes the signal, there is a cutting of the RNA. And so this signal is recognized by certain proteins so CPSF is involved in cleaving the RNA. There's a cleaving stimulation factor. Then there are other cleavage factors that are shown here. So all these proteins come together to determine that this is a place where the messenger RNA should be cut. This is the end of the messenger RNA, and this is just the RNA polymerase continues to transcribe until transcription is terminated. Now, once that is cut, what polyadenylation is that there are enzymes. There's an enzyme that will add adenine residues onto the 3' end of the messenger RNA, and it will add many adenine residues. So that is catalyzed by this protein sort of highlighted in blue here, polyadenylate polymerase or poly A polymerase. And then there is also polyadenylate binding protein, which binds and stabilizes this interaction. So this is added onto the end of the messenger RNA. Now this portion again, this portion of RNA is not important. This is just the continuation of RNA polymerase with transcription until it gets terminated. So up to 250 A residues can be added to the 3' end, and this is important for export of this messenger RNA out of the nucleus and also for stability. So if we look summarizing up what has happened in eukaryotic and prokaryotic mRNAs and what they look like. Here's our eukaryotic messenger RNA. It has this 5' cap. Now there are regions that we didn't talk about, but there are untranslated regions. So if they're located on the 5' end, they're called 5' untranslated regions. If they're located at the 3' end, you guessed it, they're called 3' untranslated regions. And these can have sequences that are important for regulation of this messenger RNA. They don't encode protein. They're untranslated. Called that for a reason. The translation for this protein would start in this darker blue portion, and a protein would be made from this portion of the RNA. And then these mRNAs have poly A tail on the 3' end. If we look at prokaryotic messenger RNAs, they don't have this 5' cap. They do have these 3 phosphates. They also often have untranslated regions both at their 5' and 3' ends. A major difference is that you can have more than one coding sequence on a prokaryotic or bacterial messenger RNA, whereas for eukaryotic messenger RNAs, there's typically only one coding sequence. And these coding sequences in bacterial messenger RNAs can each be translated into different proteins. And you also notice that there is no 3' tail, poly A tail on bacterial messenger RNAs. Okay, so if you look at table 12.5, there's also a lot of information sort of summarizing this. So the last part I want to talk about in this is mRNA stability. So the stability of eukaryotic messenger RNAs varies considerably. It can be as short as several minutes to several days or even months. And so the stability of a messenger RNA can be regulated so that its half-life, how long it's around, can be shortened or lengthened. And this will influence the concentration of this messenger RNA and, as a consequence, gene expression. So there are some factors that can affect RNA stability, how long the poly A tail is, and what destabilizing elements exist in that RNA. Okay, so I have this very complicated, messy-looking slide, but I'm going to go through this, and it's not too bad. But this shows the different ways that a eukaryotic messenger RNA can be degraded, and so how this is so where what parts are giving stability to the messenger RNA, and when those are compromised, how does the messenger RNA get degraded? So I'm going to talk about these two outer portions first, and then we'll talk about the middle one. So here's our messenger RNA. We have our 5' cap, our phosphates, our start codon and stop codon for the gene, our 5' and 3' untranslated regions, our poly A tail, and poly A binding protein bound to that poly A tail. Okay, so that's what we have going on here. So there are enzymes in the cell which can remove this cap. They are themselves regulated, and so this is called decapping. So here we have our little Pac-Man here coming along and removing this cap. Now what happens then? So I mentioned that this structure of this 5' cap, being that it's a 5' to 5' linkage, is different from what you would expect on a messenger RNA. This is not recognized well by lots of exonucleases. However, once this is removed, having a sort of normal 5' end can be recognized by 5' to 3' exonucleases. These are enzymes that can bind to the 5' end of the messenger RNA and start chewing away nucleotides, moving from the 5' end to the 3' end. So the messenger RNA can get degraded. So what happens on the other end? There are also enzymes all the way over here that deadenylase enzymes that can degrade the poly A tail. So most of the poly A tail is gone at this point, and it started out like this. So when that gets removed, that poly A tail, this becomes the target of another kind of exonuclease, a 3' to 5' exonuclease called the exosome. It's a multi-subunit protein. And this exosome will degrade the messenger RNA coming from the 3' end. So these components here at the 5' end and 3' end, the 5' cap and the poly A tail, help to protect the important part of this message. So if they get compromised in any way, this message is now susceptible to being degraded. So what's happening here in the middle? So what if we've talked also about other kinds of enzymes called endonucleases, which cut in the middle of a DNA or an RNA. So this would be an RNA endonuclease that happens to cut in the middle of this messenger RNA. Well, now what goes on here? Now, the messenger RNA, this 5' end is still protected. This 3' end is still protected. But look at what's happened here. For this portion on the left, there is no protection on the 3' end. So this 3' end looks like this 3' end, and the exosome can degrade this. Now, this right-hand portion of this messenger RNA now has no 5' cap. So this looks like this guy over here. So this 5' to 3' exonuclease can degrade this portion of the messenger RNA. So we can have degradation of this of a messenger RNA that's cleaved by a nuclease by both the exosome and a 5' to 3' exonuclease. Okay, just a little note about the exosome. This is the last thing to mention here, that this is a multi-protein RNase, and here's the structure of it. And you don't need to know all these components here, and that its activity is to degrade messenger RNAs in the 3' to 5' direction, and degrade messenger RNAs that are lacking a poly A tail. Now, the thing that's important is that this is an essential component, this exonuclease, so it performs an essential function, and it has been implicated in autoimmune and motor neuron diseases. Okay, so that's where I'm going to stop, and we'll pick up with the next lecture soon.